Well, as we continue with this meeting, we appreciate everybody staying with us and coming here today. And again, I'd like to thank Seth for his generosity to allow us to be here. Sometimes we have to stop and reflect a little bit of what SARC is. And many of us, as you know, from 2003 have been here. And things have changed over the time. And one of the things that's changed is that we do have a chief medical officer, and that's myself. I'm Scott Okuna. I'm a medical oncologist, sarcoma doctor at Mayo Clinic Rochester. And we thought it'd be worthwhile just spend a little bit of time re-educating all of us of what some of the things we do at SARC and how we can engage all of you that are in the room. We'll spend a little time talking about how people engage with SARC. We'll also discuss the Clinical Trials Forum. And the Clinical Trialist Forum is a unique opportunity for investigators to meet between meetings. And then we'll briefly talk about some thoughts as we continue with international collaboration. We're very fortunate to have meetings twice a year. The meetings here at ASCO are great. It brings everybody and sets the tone for the rest of the week at ASCO. And we know that CTOS is a great opportunity because all sarcoma people go to CTOS. Twice a year is probably not enough to keep people informed of what's going on and keep things moving. So we do have existing methods that all of us can continue to connect between our meetings at ASCO and CTOS, and that includes the newsletter as well as podcasts, and we'll get into that a little bit. We also felt as though that there is a missed opportunity to continue to update where people are with the active clinical trials that we do have. We also want to let people know what trials are actually in development and also give investigators the opportunity to pitch ideas, to get feedback from a group of clinical trialists. So out of that, we did develop what we would say is the SARC Clinical Trialist Forum. And we meet probably 4 times a year virtually. And we do have an agenda that's set that kind of discusses those. The Trialist Forum allows us as sarcoma providers, clinical investigators, and researchers to continue the dialogue of what we can do to improve clinical trials and research that we do at SARC. These are not only for SARC studies, but also raises the bar for all of sarcoma research. So that is the advantage that we do have. If you are interested in being on that distribution list to get that invite that comes out, please let us know. As we said, we're very blessed to have Kim Ward, who is running around here somewhere, but her email's here. You can also go to our website as well, and there's a link there for the SARC Central Office, and you can ask to be put on that mailing list. We do send out an agenda. We do have dialogue that goes on. We do keep a little bit of minute meetings, but we kind of update people here at our major face-to-face meetings. So hopefully, if you are interested, that gives you an extra opportunity. We also realize that we have been focusing heavily on understanding how we can do clinical trials here in the United States and certain collaborations outside the United States. We know that SARC is for everybody. So if you want to be a participant in clinical trials, research, or activities, anybody, US or non-US, can be involved. We have to be very strategic as we go into the international community. Why? We have a hard enough time navigating the US infrastructure. In fact, I have a hard enough time understanding Mayo Clinic, so that's a very big deal. So as we start to partner, we do know that logistics, getting drug to and from, the expertise of each of your research institutions to do trials, and then all the legalities as crossing borders is huge. Have we done it? Yes. Can we do it? Yes. Will we continue to do it? Yes. And we're very fortunate that some of our major studies have included cooperative groups outside of our standard US-based groups. So I listed some areas that we want to do relationship with or have done relationships, And these are not the end-all be-all, but if you are interested in helping this international effort, I think we would be encouraged to kind of figure out how to do this. We have started some dialogue with some of the groups in Canada. Why Canada? Well, they have an organized system up there. We don't know that system, but we know the people how to get into there. So as we start to think about trials, we can probably work and develop ways that we can work with the existing structures that are available in Canada. We've done the same thing with Australia with some of our studies as well. We engaged groups of people that already knew those areas. So as our map kind of shows, we have a lot of sarcoma centers that have high volume, that have expertise in treating and managing patients in a multidisciplinary approach across the world. Yes, we have a lot of dots, as we would say in the United States, but we have a lot of great dots and a lot of great investigators and people in this room and also on the map that are not in the US. So let's continue this dialogue. So if you are interested in helping be part of a group as we continue this dialogue, please send me a note. We'll continue to formulate how we go about doing it, and your input as we interact with different groups outside the US would be critical. So with that, I'm going to introduce our next speaker, and this is Dr. Cote. You want to come up here, Greg? I'm going to introduce both these individuals here. Greg Cote is the chair of our Clinical Trials Development Committee, and Libby Davis is the co-chair, and they're going to share a little bit about ways to consider clinical trials.
All right, thanks Scott for the introduction, and thanks to all of you for your attention for the next few minutes. Greg and I just wanted to highlight what we're doing with the Clinical Trials Development Committee. So this is the SARC organizational structure. I know Scott thought it would be good to kind of reintroduce everyone to SARC, and we thought it would be good to kind of show where we fit into the organizational structure. So at the top of this diagram is the SARC Board of Directors, followed by the CEO, the CSO, and the CMO. We are, I don't know if this has a pointer, but we're kind of down there. All right, we're down here in the bottom, kind of underneath the CMO. We oversee also the local control working group and their concepts and try to help steward them through the process. So our mission is to review and develop clinical trial concepts. We also mentor early career and clinical investigators. So we've had some early career investigators with some ideas, but just needed some guidance or some more senior investigators, maybe outside their institution, to help them with their concept, and so we can help pair people. We also foster strategic relationships with industry partners. And then we also assess all requests for use of SARC's clinical trial data as well as the samples. So this is the current membership composition of our committee. So we have a chair and a vice chair, Greg and myself, as well as a SARC CMO, COO, a statistician, and then we have medical oncologists as well as either a med-peds or an AYA oncologist. And we've elected to limit this to one member per institution.
All right, thank you. All right, so who's on the committee? So we have your chair and vice chair, as you heard. Scott is our CMO, also part of this. And the three of us represent what we call the executive committee. And then Claudia is our COO, as you heard earlier. And our statistician is Carla Ballman. Many of you know her. She's from the Mayo. She provides statistical support. And then our panel of medical oncologists include Drs. D'Angelo, Armstrong, Bowie, Manga, Razik, and Palmerini. And our med-peds oncologist is Dr. Livingston. So you may recognize some of these members were on prior iterations of this committee, and then there's some new faces as well. We had a call for volunteers a few months ago to help build this committee. If you missed that and you're interested, let us know. We kind of envision this as a committee where people will kind of come and go over years as their schedules get busy. They may rotate off, and we'll be looking for new members. I'm sure if you're interested in doing something now, Scott probably has many roles to fill. So just please let us know. Okay, so I wanted to spend a minute and just talk about how concepts go through our committee evaluation. So whether it's an internal concept that was developed by somebody on one of these SARC committees, or whether it's an external concept from one of you, this will go to the executive committee first. This is kind of like a thumbs up, thumbs down kind of thing. We'll give you some initial feedback, but really we just want to make sure it's within scope. Then we're going to link you with a couple members from the committee who will meet with you separately, go over your concept, provide feedback as needed, and then we'll bring it to the larger committee. For industry partnerships, these are really driven by relationships with Steve and Scott. So they're going to take the initial lead here. And then as those mature, we'll bring it to the other committee members. All right, so what are our goals? So we've set some short-term and long-term goals that we're working on building through. As you heard earlier, we're trying to open 3 to 4 trials per year. We realize this is ambitious, but we think we can do it. And then we really want to support individuals and sarcoma centers that are submitting proposals. This could be at various different levels. As you heard, mentorship is really important to us. That was something that was important to Brian that we're trying to carry on. So if you're an early career investigator, we can link you with a mentor. If you need help with protocol design, finding funding, budgeting, stats, these are all roles that we can fill to smooth this process. We're also working on refining our submission and review process. And right now, I kind of gave you the highlights of that, how we're working through that, but we're working on trying to make this more efficient and more constructive. So that's a process that's ongoing. And then importantly, we also want to reevaluate our site selection process. We see kind of 2 competing interests here. So we want the wider community to be able to participate in studies, but we also need sites that are going to accrue. So these are 2 competing interests that we need to balance, and we're reevaluating that process as we speak. And then as Scott just mentioned, one of our other goals is really to strengthen these non-US partnerships. You're going to see some examples of that as we go through some of the existing and pending trials. So that's all I got. We're happy to take any questions if anyone has any.
Any questions for Libby or Greg?
All right.
Very good.
All right, thank you.
So our next area is we're going to be discussing some new program announcements, and Steve will have you announce.
Thank you, Scott, and thank you for what has been an extraordinary morning already. What we're going to talk about briefly, thank you. We're announcing today the launch of a new sarcoma symposia series that's going to be embedded within the SARC semi-annual meetings and potentially in the in-between quarters when we're all not getting together as a group. So very proud to announce the launch of the Chalo-Luskin Sarcoma Symposium Series. I'll talk about what it is in a second, but in the meantime, first want to call out the benefactors. Renee and Maya Luskin, who some folks may know are rather extraordinary philanthropists who have supported UCLA in particular with many incredible structures built up. Thanks to the connection with Dr. Sant Challa and his lovely wife Mihir, we were able to secure a sponsorship to support this. And this is important because getting many of you folks to go to these conferences can be expensive, as you know. We already talked about our Young Investigator Travel Award. This will allow us to get cutting-edge folks into the room that may or may not normally come to a SARC, to a CTOS or an ASCO meeting. The topics developed will be developed by SARC's leadership in partnership with the selection committee with, and help in a minute, Dr. Arun Singh. And the inaugural symposium will be held actually at CTOS in Seoul later this year during SARC's normal 3 to 5 p.m. Normal because we're very grateful to CTOS leadership for permitting us to do this. And Dr. Singh, are you nearby? Yeah, please come on up. He was going to just say a few words, and after that, we're going to move on to the rest of the agenda.
Thanks, Steve. So how I got involved in this endeavor is Steve was visiting Los Angeles. We started talking about the sarcoma field, and we really tried to focus on what things need to be done to have the greatest impact in improving the care of sarcoma patients. And we focus on 2 main themes, and themes that I think you're all very well aware of: drugs and money. That's what's necessary to make sarcoma better. You need resources for trials and science, and we really need novel therapeutic agents. When you think about the latter theme, when you look around to see where that's been done well, if you have a bird's eye view of the work that Dr. Sant Challa has done over the course of his career, there are some things he's done incredibly well. One has been able to access novel compounds, and the second thing has been extremely creative in using the drugs that we already have available. So either through drug repurposing and also through coming up with novel clinical trials. And as you know, he every time you come to SARC or CTA or to ASCO, he publishes several trials, and he's put a lot of patients on clinical trials, which is part of the goal of all what we're all doing is really to bring novel drugs to patients. And so that brings us to the heart of the Chawla-Luskin Symposium. It's really to invite scientists to come and talk about new areas of molecular biology or other fields we're really not familiar with, to hopefully spark some new ideas or to give us some other ways of thinking about the sarcoma problem. At UCLA, you can't walk around without seeing the Luskin name. In Santa Monica, you can't walk around, or SARC, without seeing the Chawla name. So it's fitting to have the Chawla Luskin Symposium. And I want all of you to join me in thanking Dr. Sant Chawla and the philanthropist Meyer Luskin for their generous gift. Thank you.
Thank you, Arun. And it's a great opportunity and great collaboration with a lot of great people to help continue to move the science along and get awareness for sarcoma. In that vein, we like to kind of highlight one of the initiatives that was started several years ago is SARCTALK. SARCTALK, our tagline is interviewing interesting people who are connected to sarcoma, where we can all share and spread the knowledge about sarcoma. And I think that is our main goal at SARCTALK. We're in our 4th season, and for those that don't know, SARCTALK is a podcast. We also have a video component with that you can watch, and the 4th season started. We started in November of 2022. It's hard to remember, November 2022. Bill's here, Bill Tapp, along with Brian Van Tine, were our 2 first guests at that time. We've recorded 39. As of today, it's 40 episodes. Many of you in the audience have been interviewed because you are interesting people connected with sarcoma. But we want to expand that as well. So with that, we have 2 initiatives that we have started. One is called SARCTALK Journal Club, and the other is Patient Experience, Rise to Face the Sun. And I'm going to highlight something that we're going to be starting. Catherine Wilbur, who is a medical oncologist at Johns Hopkins, she's not here today. She had some events. She works with Christian Meyer and the whole team over at Johns Hopkins. Her and I will be co-hosting SARCTALK Journal Club. We actually did record a podcast about that. We're going to be basically presenting opportunities for discussion of recently published sarcoma articles. Our goal will be looking at the clinical ones first, and our plan is to review probably once a month. We'll record a little bit more often, less often, but we'll post about 1 journal club a month. That way we can keep all of us on task because we're all busy. Some of us have our own journal clubs at our own institutions. However, this will give you an idea of how at least Catherine and I think about the journals that are published. And then what we're going to require of you all that listen is to send comments. Because then in follow-up recordings, we will highlight the comments because we might miss the mark. I can tell you there are times when I have missed the mark, when we have all missed the mark, and you all can keep us on task. So if you are interested in submitting articles or potential articles, Catherine or I will be happy to review those. We might not get to all of them because there's only time constraint, but this is a great opportunity to continue to share knowledge about sarcoma in high clinical articles. With that, we also want to highlight a new initiative that was started, and this is the Patient Experience, Rise to Face the Sun. This is started by Chappie Conrad, our previous board chair, and he and Daphne got together and said, hey, we need to get the voice of a group of people. And with that, we've been blessed to have Daphne working with us, and I won't share a little bit too much because Daphne is here today. She's an outstanding, beautiful woman who has a vision of how to connect to different individuals in a very kind and caring way. So with that, I want to introduce Daphne Egren, and she will share her next series of podcasts. Daphne?
Hi, thank you. Okay, awesome. So just to talk a little bit about what this podcast is about, and then I'm going to go into who I am professionally, personally, how it connects with the podcast, and then we're going to talk a little bit more about the podcast. So this initiative came out of wanting to do an awareness campaign for sarcoma, because it's rare, not a lot of people know about it, and more people should know about it, right? And what's kind of weird about doing an awareness campaign for such a rare cancer is, how do you actually get people to care about something that they don't really have a lot of connection to? So that's one thing. But another thing is, and because it's rare, you have a lot of patients from like all around the world who are, connecting with each other over social media, which I learned very quickly as I was diving deeper into this. And so I decided, let's just do a podcast where we interview these people who've either battled sarcoma, battling sarcoma, or even caregivers like moms or even siblings of those who had sarcoma, and just ask about their experience. And then I always ended it with, what advice do you have for people who are now encountering sarcoma? And what's nice is that we've gotten a lot of different kinds of people. So people who are encountering sarcoma for the first time, hopefully they can find someone that they feel a connection with and a story with. We have 10 episodes that are coming out, and I'll talk about that just in a little bit. The first 2 are actually released today. So who I am professionally. So I'm actually a senior medical writer here at Northwestern Cancer Center. I write for a lot of different protocols, so breast, lung, skin, et cetera, but I'm lead for sarcoma and for neuro-oncology. And if you don't know what a medical writer is, because I recently learned that not every institution has one, basically, principal investigator will come up to me or one of my colleagues with a concept or a grant, et cetera, and we will help them write a protocol. I'm also a student at Harvard University. I'm doing my Master's of Public Health right now, and that's me with my classmates. And so that's a little bit about me professionally. But to get a little bit more personal, because this conference room and I have a lot of history, actually. So this is me. About 11 years ago, I actually moved from Colorado, which is my home, to Chicago to start at School of the Arts to Chicago. If you don't know what that is, totally fine. It's an art conservatory, and I had been working since a very young age to be a fine artist. I got this big scholarship. They got me a grant to study in Italy. It was really cool. But first week of moving here to Chicago, I got diagnosed with acute respiratory distress syndrome. I arrived to the hospital on 50% oxygen. I was on the lung transplant list. I was placed in a medically induced coma within 24 hours of arriving. I was on ECMO, I was intubated, and I had a 2% chance of survival, and with that, they thought I was going to have brain damage. So I was in a coma for about a week and a half, something like that, and in efforts to try to get me off of the lung transplant list, they said, let's have you walk on ECMO, which at the time, this was in 2014, was not super common. I mean, it had happened, but wasn't really that common. And here's me in the middle, and that was the first time I walked on ECMO. I did it for a few times after that. First time I walked on ECMO, actually after being in a coma for a while, I walked for 30 minutes, and I didn't get it. There's all these Northwestern students watching me, and I remember just not understanding what the deal was. But I became the first patient at Northwestern and in the state of Illinois to walk on ECMO. This is not very common for, someone at, that age, 18, to have ARDS. So it was actually a very lonely experience afterwards because I didn't have anybody to connect to, which actually leads me to the podcast that I'm doing. But the care team that I had never experienced that kind of compassion before. And I just thought, I'm going to get back into that hospital. So a month after I was discharged, I had made everybody, like the main people on my team, pieces of artwork. And I went back to the ICU and I delivered it to them. And then I approached one of the main doctors on my team and I said, hey, I'm an artist, but I'd like a job here. That's actually how it happened. And he said, okay, do you want to be a wet lab research assistant? And with a tremendous amount of excitement and a healthy amount of fear, I said, yeah, what's that? And so I found out I was actually very good at it. And so you can see at the very, over here, that's my art school hybrid wet lab research assistant. And then I worked in research all the way up until now, after graduating Bachelor of Fine Arts as well, because I continued to do art school. But so then I became a senior medical writer, right? And primarily in sarcoma. And as I was, learning more about sarcoma, something that I was reading about is that it mostly, it affects a lot of adolescents and young adults. And I thought from my experience, I had never met somebody else who had what I had at that age, and you're prepping to, start your, education, your career, what have you. And no one tells you, just be careful, you might get something like sarcoma, because that's not exactly, how we talk. But it happens, and it affects a lot of people. And so these are a few of the people that I've interviewed so far. I actually have interviewed more people since. We have McKenna, who's jumping in front of the pyramids. Brandy Benson, who's going to be presenting at the very end. So I don't want to say too much about her, because she's going to be talking lots about her experience. We have June, who's from Canada. Valerie, who speaks on behalf of Josh. And then Julie Kramer, who's with the Synovial Sarcoma Foundation. So those are a few people. And then two other people I recently interviewed. First is Gianna, who just rang the bell after finishing her chemotherapy after having 412 desmoid tumors removed from her body. So that's her. And then we also, I interviewed the CEO of Stupid Cancer, Allison Silverman, whose brother passed away from Ewing's quite a few years back. The first episode that's going to be released today is Eli. So what we're doing is we're releasing these teasers on social media, so people are aware of these podcasts that we're releasing. And on the podcast, I won't tell you too much because I want you to listen to it. He said, yeah, I have a reoccurrence of synovial sarcoma in my lungs, and I have to go and get tested to see if I'm eligible for T-cell receptor therapy. And I said to him, well, I have the highest optimism for you. I told him, I said, hey, we're releasing the teaser on this past Monday. And he said, oh my God, I'm starting T-cell receptor therapy that day. And so he's on day 3 or 4 now. I can't count. But so that's the first episode. It's going to be released. The second episode, I'm sure there's a lot of people who know who Natasha Allen is. She had a very, very bright presence all over social media, and she really wanted to talk about cancer in a much different way. We have her mother speaking on her behalf. May she rest in power. She passed away August 22nd, 2025. And before I have Anne Graham come up with her survivors, I'm just going to play a short clip from Sabina's interview, just to give you a little taste of what this is like.
I always hear from her somehow, if it's through a note that she left me for her birthday, on her birthday, or if it's hearing from you. So she said, so she left me this note on her birthday last year. Obviously she wrote it before, I just happened to find it that day. I'm sorry, not for what I have done, but for the inevitable. I wish the weight of mortality didn't have to burden you. No mother should lose a child. And for me, and for that, I'm sorry. This disease may take me out, but I hope it doesn't dim your light. You are the reason I got the years I did, instead of focusing on what took me. Think of what kept me going, and what time I did have, the moments we shared, the laughs, the cries, the big moments and small. And we all have a limited time on earth, and I'm blessed that I spent mine with you.
Wow.
Well, as we have our next group coming up here in clinical trials update, we'll have them all come up here because we are running a little bit late and we do ask for some grace as we might go over a little bit. But we want people to come up here that are ready to present. Our first opportunity to share is Sandra here? Okay, the other, okay. The actual Dr. D'Angelo, yeah. So we're very fortunate that we do have a group of clinical trials that we want to share with you that are actually open and accruing. We want to highlight some that have been completed and then also some that are planning to launch. Sandra?
So thanks. I'm here to present on SARC 045. This is the phase 2 trial of Tebentafusp in patients who have HLA-A*02:01 positive clear cell sarcoma. Andrea Napolitano couldn't be here, so I'm presenting on his behalf. Clear cell sarcoma is a sarcoma that often can be slow and relentless, but a tendency to recur and metastasize. The prognosis is quite dismal. It's characterized by a specific fusion, EWSR1, ATF1. It stains positively for numerous melanoma-specific antibodies, including HMB45, MITF, and Melon A. There's no agreed standard of care. The median progression-free survival is quite poor, 2.5 to 5 months, highlighting the need for innovative and new options. Tebentafusp is a soluble T-cell receptor that's fused to an antibody, single-chain variable fragment. The target for Tebentafusp is GP100, which is presented in the context of MHC class 1, and it's designed to activate T cells and is in physical contact with nearby CD3 positive cells. Tebentafusp is actually FDA approved for uveal melanoma. The most common adverse events patient experience with this drug is rash, fever, pruritus, or hypertension. The most common grade 3 events is also rash and elevated transaminitis. Typically, these events are most common within the first few weeks of treatment and often decrease in severity over time. The goal and the primary objective of this clinical trial is to estimate the progression-free survival of patients at 24 weeks who receive this agent. There's numerous typical standard secondary objectives. Inclusion criteria, most notably, is the need to have HLA-A*02:01 positive status. Patients must have appropriate performance status. We do require a progressive disease by RECIST 1.1 within 6 months of study enrollment, but not for those patients that are newly diagnosed. These are fairly standard exclusion criteria. In the interest of time, I'll try and move things along a little bit quicker. So this is an open label phase 2 trial. The drug is administered weekly in escalating doses. Initially, there's a step-up dosing where patients receive a portion of the dose on day 1 and then on day 8. And typically then it's every 3 weeks thereafter. Scans are performed at week 6 and every 6 weeks thereafter through week 48, and then every 9 weeks thereafter. For those patients that are enrolled to receive Tebentafusp, we do require mandatory biopsy at baseline and on treatment, as well as serial blood collections. For those patients that don't have the appropriate HLA, they have the option to enroll in a prospective trial where we're able to understand the natural biology and responses with essentially investigated choice drug therapy. These are the study sites. We're currently open at MSK and at USC with Dr. Igolnik. Pending opening is Northwestern with Dr. Pollock, as well as the Royal Marsden with Dr. Napolitano. There was 1 patient enrolled, and there have been 5 patients pre-screened, so we just want to put the message out that these studies are open, at least in the east and west coast of the United States currently. And if you screen patients actually locally, we can see patients and consent them on the same day and really try and make the process a little bit more seamless. And these are the contact information. Thank you.
Thank you, Sandra. The next study is SARC 46. Dr. Wagner?
All right. right, so thank you for the opportunity to highlight this trial again. So again, for the sake of time, I'll try and speed through. Much of this is exactly the same as what I presented a year ago. So this is a phase 2 study of Nab-sirolimus in patients with progressing or symptomatic epithelioid hemangioendothelioma. So I think most of you know what epithelioid hemangioendothelioma is, but it's a very rare growth of endothelial cells and characterized by fusions involving the hippo pathway genes. There has been primarily retrospective study, especially out of the group in Milan, showing that sirolimus has activity in this disease. The largest cohort published to date was 38 patients, all of whom had progressing disease. And in that study, sirolimus had a median PFS of 13 months with a response rate of about 10%. The primary endpoint of the SARC trial is objective response rate by RECIST 1.1, and a number of exploratory endpoints. I think notably, we've embedded quality of life and PROs. The inclusion criteria, at least the selected ones you can see, I think key ones are that patients, of course, have to have EHE that is either symptomatic or progressing, and we leave that up to investigator discretion to define. And you can see the lab criteria and the usual performance status criteria. For exclusion, I think some key things to note that are not exclusionary right now are prior sirolimus exposure, and that's because there's some evidence, especially on the expanded access program of Nab-sirolimus for PCOMA, that patients who had previously been exposed to sirolimus could still potentially respond. And then also, as of right now, at least liver transplant's not an exclusion, although if you're going to discuss this trial with any patients, I think it requires a very serious discussion whether patients who have prior sirolimus exposure or who have had a transplant are appropriate for the study. The design is a pretty standard Simon stage 2 design with the first stage of 12 patients. If there's one response, then we would open up to a second stage with an additional 25 patients. The treatment is Nab-sirolimus, which is an IV infusion on days 1 and 8 of a 3-week cycle. I pretty much just said this. We are very actively opening actually right now. So we opened about 2 months ago at Dana-Farber, Memorial Sloan Kettering, Stanford, and WashU just opened and activated within the past few weeks, and Colorado is not that far behind. And then we're also hoping to open at MGH. Importantly, just for confirmation of diagnosis, we're working with the pathologist at the Brigham, especially Dr. Papke, who is going to review archival tissue. The status, like I said, is now open. We've already consented 4 patients, and as of today, 3 patients have been treated. And that's the summary. Oh, and I should also very importantly note, so the study is funded by the U.S. Department of War, actually now, and I'm serious, and we are very grateful to Adi Bioscience, who is supplying drug, and of course, academic assistance.
The next study that we are presenting is SARC-44. Dr. Haddocks? Well, Dr. Haddocks is coming up here just to remind everybody that Mark Dixon is in the plenary session for SARC-41. So come to that session and hear the results of Abemaciclib versus placebo in DDLP.
Good morning. It's good to see everyone. So it's no news to this group that TKIs have revolutionized the treatment of GIST, but almost inevitably, resistance does emerge, and that's often in the form of mutations in KIT ATP binding pocket or the activation loop. Bezuclastinib is a selective type 1 KIT inhibitor that inhibits clones that carry the AL mutations, and that's complementary with sunitinib, which has activity in the ATP binding pocket mutations. This combination was evaluated in the phase 3 study PEAK, which will be presented at ASCO this year by Dr. Andy Wagner. So please tune into that or come in person. And of course, that showed really promising activity, as you'll see. This study, SARC-44, is a single-arm study, which is meant to look at the combination in later lines of therapy, so patients that have already progressed on sunitinib, with the primary objective to estimate the PFS in these patients in later lines, and also to take a deep dive into mechanisms of response and resistance. Quick overview of the study design. We enrolled 40 patients. Patients start with bezuclastinib alone for 2 weeks, and then add sunitinib, and then continue combination until progression or unacceptable toxicity. We have ctDNA collection throughout, as well as patient-reported outcomes surveys throughout. And then in a subset of 20 patients, we performed a series of PET-CT scans. These were done after washout, after the 2 weeks on bezuclastinib alone, and then after the 2 weeks on the combination therapy. And that series of scans is analyzed in real time, and then we select a lesion to biopsy that's most resistant to therapy. And then that sample is subjected to a lot of downstream testing, including sequencing and looking at the KIT activation status and downstream pathways. We're also working with our nuclear medicine collaborators to look at some interesting nuclear medicine readouts and how those may be predictive of response or resistance. We are open at 4 sites shown here, and we did complete enrollment in October of 2025. We still have 11 patients remaining on treatment, and our tentative plan is to present the clinical results as well as the correlates at CTOS this year. So please stay tuned. And with that, I'll conclude, and a strong thank you to all the patients that participated, the caregivers, loved ones, all the study sites, and the research teams there. SARC, thank you so much for all of your support on this study as being the organizing center, and to all those who provided funding. Thank you, thank you.
Thank you, Candace. We have 2 new studies that are planned to launch. As you heard from Greg Cote and Libby Davis, our plan is to do 3 to 4 a year. As you know, it takes years to get these in development, and we're very fortunate for the first one is Dr. Liebner from Ohio State University, SARC-48. And it's this close.
Awesome. Thanks, Scott. We've been working very hard to get this one open, so I'll provide some updates on this one. The study title is comparing treatment with or without a neoadjuvant doxorubicin and ifosfamide in selected patients with high-risk resectable soft tissue sarcoma. That title was mandated by the study funder, but it doesn't fully encompass what we're hoping to achieve here in this study. So I think, as everybody is well aware, SARC-32 really has changed the therapeutic landscape for UPS and extremity LPS. So this is kind of the study design for SARC-32. I think most people are fairly familiar with the study, essentially demonstrating improvements in 2-year disease-free survival in patients who received neoadjuvant pembro RT and adjuvant pembro compared to patients who received radiation, neoadjuvant radiation, both arms receiving surgery, of course. And so there was a pretty significant improvement in outcomes in terms of recurrence risk, the hazard ratio of 0.61 in the published article. And so the question obviously is, how do we treat our patients now? And I think a lot of folks have kind of looked at the data and kind of been trying to understand how we think about conventional cytotoxic chemotherapy in these patients. The current, most recent version of the NCCN guidelines does list AIM or similar regimens and pembro as appropriate for these patients.
But I think there's been some debates here at CTOS, at SARC previously about how we think about these regimens and how do we incorporate in our patients. So, we have good reason to believe that chemotherapy is still appropriate. There may be some synergy with immunotherapy. It may be complementary. And we hypothesize that there still is a role for conventional chemo in highly selected patients. I won't go into all the background data here, but I think there's a lot of folks who are digging into those questions about what patients might benefit. So we actually reached out to PCORI. I don't know if how many folks here are familiar. They're essentially, their primary purpose is to fund comparative effectiveness clinical research. And so essentially looking at cases where there's some ambiguity in terms of how patients are approached in the real world and trying to provide some additional clarity. Importantly, and this is important for the study design, it covers research costs but does not cover patient treatment. So it was selected for funding. We are, as Dr. Kuna said, this close. We are kind of working our way through the final details of the contract. But this study essentially is intended to look at the impact of 3 cycles of neoadjuvant AIM in patients who otherwise meet SARC32 criteria, and comparing that to patients who receive the standard SARC32 protocol. So all patients will receive, let me go back. So patients randomized to Arm A will receive 3 cycles of AIM chemotherapy followed by pembro, radiation, and surgery per SARC32. Patients randomized to Arm B will receive pembro, RT, and surgery per the SARC32 protocol. The primary objective is similar to SARC32, it's disease-free survival at 2 years. And we believe we have roughly an 80% power to detect an improvement in relapse-free survival of 0.6 comparing the 2 arms. Secondary objectives are, we're including quality of life, overall survival, local and distant disease-free survival, and of course, importantly for this is treatment-related adverse events. And so we do have several exploratory objectives that we're looking to. One of the main things that we're, exploratory objectives are not covered through the grant mechanism, but we will be collecting biospecimens, and we're looking forward to using those to try to answer some important questions in these patients. Key eligibility criteria, adult patients, tumor size greater than 5 centimeters. We are trying to predominantly focus on grade 3 because that does appear to be the subtypes where there is the most data supporting the use of cytotoxic chemotherapy. Grade 2 are permitted if their FNC-LCC score is 5 and there's clear necrosis on imaging or biopsy. And otherwise, patients need to be eligible for definitive local management with limb salvage. Study sites. So we did send out a feasibility assessment last year, thanks to all those who completed those. As we get closer, I know SARC's reached out to several sites just to kind of make sure that we're all aware that we are very, very close. Priority sites have been identified, but as we have not finalized with circulation of the protocol, we welcome sites who are interested to reach out. The email is there. So please, if you're interested, reach out via sarc48@sarctrials.org. The contract is extremely close. This is not a grant, this is a contract, so they have been looking through that with a fine-tooth comb. Protocol is final, we're IND exempt, WCG is the IRB of record, and I mentioned the site selection. So I did neglect to mention that this is a fairly large study, so 228 patients. So this really is a collaboration across many, many sites. So thank you very much to PCORI, to SARC. This would not have been feasible without SARC, and the Sarcoma Coalition, who've provided a lot of feedback in terms of study design, in terms of patient advocacy. So with that, I'll conclude. Thanks.
This will require a national effort to get this study accomplished. It has to be completed within 5 years. The last study that we're going to present that hopefully launch in 2026 is be presented by Dr. Herbie, SARC49.
All right, I'll try to speed. So just in a way of background, we know MPNST are terrible. The median PFS in all trials to date has been only 1.77 months, although I will argue that no trial to date has been stratified by any kind of genomic or otherwise biomarker type of predictor. There is preclinical data showing that MET amplification is an early occurrence in MPNST progression. Some of this data is from Kayla Torres's group. Some of the data is from Matt Steensma's group. MET inhibition has shown some activity in MPNST models. And importantly, p53 loss alters MET dependency and leads to therapy resistance. So there is some data here showing in the heat map pretty high expression of MET, and then data from Matt Steensma's group. So the blue bar is the p53 wild type MPNST that are treated. You can see a good response, but not a good response in the p53 mutant tumors. Zanza is a multi-receptor oral tyrosine kinase inhibitor, hits many oncogenic targets, including MET. So our hypothesis is that Zanzalintinib will have clinical activity in p53 intact MPNST. So this is our schema. Again, patients with advanced MPNST, they have to be p53 wild type, ECOG 0-1, any line of therapy. They'll get Zanza daily. Our endpoint is 4-month PFS. If we can hit that, that would be amazing for this sarcoma. If we do, we'll enroll an additional 12 patients, and again, they'll continue until progression or unacceptable toxicity. So it's a Simon's 2-stage design, multi-center open label study, again, Zanza and p53 intact MPNST. Patients have to be 18 or older, and our goal is 22 patients if we complete the second stage. Again, our primary objective is 4-month progression-free survival rate. Secondary objectives, overall survival, overall response rate, durability of response, and the toxicities that we see. We will need archival tissue for sequencing. I'd like to correlate response to genomics and MET levels. We'll also be collecting ctDNA pre-treatment, day 1 of each cycle, and at progression that Jack Shern, NCI, and I will, and Taylor Sunby will analyze. So again, 18 years or older, histologically confirmed diagnosis of NF1 or sporadic, p53 wild type MPNST that's unresectable or metastatic, at least one site of measurable disease. They can't have Zanza in the past. Brain Rets are okay if adequately treated and stable for at least 4 weeks prior to the first dose. Study sites will be at UAB, WashU, Hopkins, and NCI. Dr. Yulo is the PI. I'm co-PI with her. Exelixis is funding this study. Their protocol review meeting is June 25th, but Vanessa and I have finished the protocol. We're in the process of building the CRFs and the REDCap database, and our goal is to open in fall of 2026. So thanks to Exelixis, SARC, our NCI collaborators, and my division that's letting me eat the cost of CRFs and REDCap. So that's good.